tracks of variation multiplied by individual scores of the patient. And so at the end of the day, you will summarize these patterns, these complicated, noisy patterns, by a few simpler patterns, and you will say for each patient, you will see what combination of this pattern that patient has. And that, as I say, our motivation came from people infected by COVID, which were followed up for, a few months afterwards to try to see how their immune system was or was not recovering. I'll show you some of the data there that our collaborators were collecting. So they had a kind of complicated setup with 6 groups, because they also did some, healthy controls for some of their, and they also did some screening just to get really asymptomatics. But for the longitudinal thing, I will just look at 4 categories of patients, which you see here, B to E. So basically starting with non-hospitalized, just patients which were symptomatic but did not need to go to hospital, and then hospitalized which did not require oxygen, were just monitored, and then you have the people which had supplementary oxygen in darker gray, and then those on ventilation, which are black. I'm just saying these colors because my plots afterwards of the score will be from light gray to black, and will follow severity. So this will be what I'll show you, the example of basically, we will reconstruct these trajectories. Of course, I will analyze only a few biomarkers, but no, there were many. So just some notation, just to give you, not to scare, but just to give you the flavor of it. So, as I say, we want the major source of variation in your data, and we are assuming that we are getting, a sample of realization over time. Time is continuous, but of course we will just get discrete. But we're making some assumption that the underlying dynamics is continuous, and in particular the mean and the covariance function are continuous. And under this hypothesis, then you could do something very similar. You used to get, when you have a matrix, you used to actually be able to compute, to decompose your matrix in terms of eigenvalue times eigenvector. Well, if you think of time, you've got loads of covariances going on between all the time points. So this is called the covariance operator. It also got a mathematical decomposition, but this time in terms of function. So it's eigenfunctions. They're kind of major, like the eigenvalue, the eigenvector represent a major, sort of. Anyway, I'm not going to do the math, but anyway, just to give you a picture, this eigenfunction are represent patterns of over time, and these eigenfunctions. So basically, what's called the functional PCA expansion, it just projects, the track, it models it as a mean plus a sum of these eigenfunctions. Very similar as a matrix, you could write it as sum of eigenvalue times eigenvector. But this is a function, and these are the scores of the particular individual I, with which says what's the importance of the eigenfunction L. So it was just to give you some idea. Of course, what you've got, this is mathematically an infinite expansion, but of course, we'll have truncated version, and of course we've got lots of noise, so we're adding, Gaussian noise to our system. So just to give you, after I'm jumping from math to biology, so just to give you an idea of what happened if you just monitor one biomarker of inflammation, which is C-reactive protein. So it's the most used marker of inflammation. And these are, you're getting basically most of the variability. You see 93% of the variability is just like a level. That's the first eigenfunction. And then you've got an interesting one which steeply decreases, and we'll interpret that in a second. And these are the score of all these patients. And you could see these are the people who are not even hospitalized. These were the seriously ill patients. And those who have basically positive score seem to be, or the positive score is severity. So basically this, you can see you're going from gray to black. So you could, and if you do a box plot of basically severity classes, and you could see this clear gradation. So basically, the first eigenfunction is a proxy for the severity. If you've got a positive thing, you're in trouble. If you've got a negative thing, you're below average. So a very neat summary of, and then if you now look at this, well, this is like this is just showing you, because you can estimate the trajectories for each patient with some confidence. So you see this, this trajectory is below the average. And this one, who is over there, is way above the average. The average is like right there. Now, if you look at the second eigenfunction, and you compare, for example, this person here to this person here, who is, who actually died, you could see that you see some kind of decrease. So its CRP level actually recovers. So having a high index, a high score on the second component means you've recovered. It's a good thing. You started high, but you've actually had a steep recovery. Whether this person goes the other way around, it's increasing its level of CRP, probably because it was catching, if you're on ventilation, you catch other infection and so on. So basically, things went crazy. So the second, so you've basically all these very noisy trajectory, you've now got a very nice summary. The first eigenfunction is the level, the second is the recovery. So it's good to be positive, shows you are recovering, and you could have confidence bands. How am I doing for time? Yeah, I think that's good. So just to say that there's so frequent, the FPCA has been around, and there's different ways to do it. If you do it in a frequentist way, you start, like the mathematics, by the covariance. So you need to estimate basically lots of covariance matrices with the data you've got, but smooth them, because you've got different time points for different people and so on. So it's a multi-step procedure where you try to estimate these large covariance matrices, over lots of different time intervals, and smooth them somehow to extract the eigenfunction, which again you smooth. So it's a multi-step, kind of computationally intensive, but it works. Whether if you do it in the Bayesian way, you forget about the covariance and you start you go from this you directly target a model of this, like a regression, basically. But because you want to be very flexible, you model them like a spline basis or something. So basically, you set up a hierarchical, a Bayesian model where you model very, very flexibly these functions, and then and you target directly this relationship. You don't extract from the covariance. That's the difference between the two approaches. So so again, this is a horrible notation, but you basically sorry. Now I just want to talk. doesn't seem to... Yeah, where I was before, I was just with one track, okay? Now I want to use several tracks together. So I've got an extra, so several longitudinal data. So I've got covariing longitudinal variable, and I'm looking at a set of them. So I've got this extra index here of which variable. So this is quite horrible, but this is the spline kind of basis. So I'm modeling a flexible mean and a flexible shape for each of these eigenfunctions, and there's a kind of simple Bayesian hierarchical model of that. Again, what's interesting is to have these scores, which can be, they have large or small, so we give them kind of heavy tail to allow for some individual to really have high scores, and so they're given heavy tail. So there's a package which we call base FPCA, which implements basically a variational. So we're not doing it through fully Gibbs sampling, but we just, again, targeting the posterior mode and doing a variational implementation. You are in a Gaussian framework with spline. It's doable. It's not. It's doable. But the interesting thing is, in this decomposition that we're using, what I forgot to really mention is that to interpret this eigenfunction, they should be orthonormal. They should be like a basis. And if you go through the covariant things, then it ensures basically that they are orthonormal. We are doing that as a post-processing. So we're doing something in a way computationally simpler. We're estimating them, and then it's a bit fiddly, but then we are making them interpretable and unique in the post-processing, not imposing orthonormality as we are estimating. It's a choice. Some people do it during the estimation, some people do it afterwards. We found that it worked well that way, and it justified. To show you, so we did quite a lot of simulation to show that it's got good accuracy, that you get some uncertainty quantification. Of course, it's variational based. You don't have the full uncertainty, but you get some uncertainty quantification. Of course, the whole... the interesting here is that the scores, they're not... There is one score per person for each eigenfunction across the p-variables. This is the basis. You have a multivariate decomposition, but it's then this you loading. And so that is good if you've got a set of covarying, if you think there's disease progression and that the p-variables, you think they're related in some way. Otherwise, you would want to do each track separately. So this multivariate version that we've got is useful for moderate set of variable which we think are covarying together, and then you want to benefit from that and summarize them with just one score. If they weren't covarying together, you would do better by having in scores doing separately each track and having separate scores, which you could always do. But to show you that here, for example, so we compared, we had six variables. One had very few data points, the others had a lot of data points. And so the multivariate version is with the bottom red line, the others are the univariate. So we're comparing basically univariate, and you could see how much you're gaining in terms of precision by using the combined with respect to particularly the first the first, which has very few values. And basically the truth is this black triangle, and it always gets covered. Of course, if you are using very independent tracks, you will, and then you are saying they have the same scores, you will have some misspecification, and you will pay some price. But maybe what I didn't highlight enough on that slide, because I didn't want you to, but basically the actual eigenfunction are variable specific. It's only the scores which are common. So it adapts to different shapes. So let's go back to 5 to end up with looking at now back to the COVID example, but we used, we were able then, thanks to this multivariate FPCA, to use jointly the CRP and 4 key inflammation and immune regulation marker, but we belong to the same pathway. So there was, we think, there was a reason that they would be co-varying and kind of co-regulated for the immune system. And this is a result then of this. So on these 5 biomarkers, these are the scores. Again, you could see that we have this looks very similar to the CRP pictures. We have the first functional. I'll show you the first functional seem to be corresponding to severity, and the second one to recovery. I'll show you the actual tracks following the biomarkers. So here we've got this. The CRP, and then these other biomarkers, the IL-5, some things which belong to this kynurenine pathways. And as you could see, we've allowed, so these are the eigenfunctions, 2 eigenfunctions. There is a level, but we have allowed different shape, and this is necessary. Even though they are related, the time shape is quite different, particularly for this one, tryptophan. And then if we take like these 2 middle, this P2, if we took the severe one, P2 and P3, you could see that basically they are, with respect to tryptophan, so P3 is here and P2 is here, we could see that they are below, either the P2 is always below the level, and this one is going down below. So being very low on tryptophan is a bad thing. And whether the others is very being high on their, having higher level, which is a bad thing. So it just, I mean, I don't have time to go into detail, but, it just captures the covariation here. You have this different thing, but you've got a single set of scores. And what's interesting is you could then export these scores, and then, you could mine it for genetics. So you've got basically captured this dynamic thing, which is a complicated shape, into simpler shapes, and then you get a set of scores, and then you could just find you could use that in a downstream analysis to try to understand. Okay, well, that's all I wanted to show you. I hope that with these 2 examples, you will be interested in marrying mathematics and basically biostatistics and biology, and thinking about structures and analyzing connections between some of the data points, and not just one at a time, or big prediction machinery. I think both are useful, but I think there's a lot of, for precision medicine, to get targets, there's a lot of interest in really trying to combine different types of omics data, contrast experiments, and so on. And, we've worked to develop flexible model, but nevertheless middle ground model. There are assumptions, there are simplifying assumptions. Of course, a Gaussian graphical model it's not, it is Gaussian at the end of the day. You're not getting the full, pictures of your data, but you're already getting where is the action in among, your many features and where you could focus on later on. And then I think approximate Bayesian inference has I think it seems just the middle ground, which we find is, after having worked in my life with full Bayesian inference for many, many years, over the last, say, 10 years, when you saw the scale of the problems coming up, I've been really converted to try to, I've kept the Bayesian modeling side, but I've really been convinced that we need to find this scalable middle ground. And whether it's EM, whether it's variational based, has a lot of potential. Again, it makes some simplified assumptions, but it buys you, a lot of nice modeling work, useful modeling work. So that's it, and I'll give you the references here of the two, the two pieces of work have been recently published, and I'm happy to take questions.
Thanks, Sylvia. Really, really excellent overview on some of those two methods. So we have times for question. As always, raise your hand, and then we'll pass on the microphone. Maybe I'll start. You show us some of the modeling about this longitudinal data, and longitudinal data are becoming more and more interesting. There are more and more longitudinal data available. You show us with few biomarkers, 4 or 5 biomarkers. Are these methods scalable when you think about longitudinal omics data? So think about multiple time point, thousands of biomarker that
There are, and I mean, there are definitely, but it could be, you need a slightly different approaches, because, as I said, you have the same scores means that you really think these things are covariant. So you sort of need to combine them with kind of a latent factors, and there are, Hélène is working on actually using that for, gene expression, but there's another twist to it. So there's a longitudinal aspect, but you do need you can't assume that one individual will have the same score for your 20,000. That would not work. So you need to add some extra. But it is Hélène and a postdoc are working on that aspect. So you could. If your scientific question is different, you want to see where are the major source of covariance, you need to maybe at the same time make subgroups somehow among your gene, and some will be uninteresting, they won't vary, you just drop those, and then you find where the action is, make subgroups, and then, so it is.
Good, excellent.
Not exactly that model.
Yeah, any question from the audience?
From somebody outside of the field, but I'm wondering, your time is absolute. I mean, there is no problem of alignment between patients and across the different P components that you look at. But what if there was sort of a different biological time between patients, so that in fact you wouldn't need also to do some alignment? Yes. I'm thinking about the second problem, not the first one. Yes. So I wonder if, in that case, maybe the Bayesian modeling is asking for too much. I mean, it's the way you presented it.
I don't know, but what you would, I mean, it might be asking for too much, but then you have this hidden, I would say, no, you have this hidden variable, which is your biological clock. You need to introduce that in your model.
Exactly.
And so can you then estimate that thing, that clock, or in a way, is there going to be one track which could represent the biological time, which will have a different role to the other one? Because,
in functional data analysis, this is the famous phase and amplitude variability. So in your case, it would be a different biological clock.
Yes.
for different patients, and maybe also for different
Yes.
things that you track. I mean, the P things that you track simultaneously.
Yes. But you could, I mean, if you have some intuition, you could try to have, a procedure which also tries to find the best alignment between your, it requires quite a lot of data, but you can and envisage that this is unknown, and then you try to learn about that. Of course, you will need some assumptions somewhere, and luck with having possibly one of your track, being the better characterization. It wouldn't have to be one, but one which would inform it more. But it is possible. Some people have done that more for, cell process and trying to find stem cells and the things which actually tracks from, development, basically. Try to understand development and where is the clock there, and realign all the different kind of, when they're just following basically expression in cells. And try to find the clock there. So that has worked in that context. But that's a specific experimental context which is more precise than,
Or maybe you could use models for the analysis that are indifferent to the alignment, so that sort of work on the equivalence classes of your functions given the alignment.
Yeah.
Excellent, thanks. I think we need to close the discussion here. So thanks again, Sylvia.